Hello. Today we are going to talk about the relationship between incidence and prevalence. We might recall from the last time that there are some differences in calculation between incidence and prevalence. In terms of the numerator, the main difference is that the prevalence accounts for all cases, old and new, while the incidence accounts only for new cases. Meanwhile, in the denominator, the differences basically lie that in the incidence rate, the time component is included, and therefore it's the sum of all animal time of observation period during the study we followed up. The prevalence value is usually useful, and it's used mainly in existing cases, as we said, and it's used for identifying disease problems for administrative purposes, because this measure, it's a measure of burden of the disease, and so it tells us how much of a disease is there. And therefore it's used for long-term disease or evaluating control strategies or defining further research priorities. Cumulative incidence, on the other hand, it's a measure of risk, and it's used to indicate the probability of individual becoming sick during a period of time. And while the incidence rate, it's not directly interpreted at an individual level, but it is an appropriate when the speed of the development of new cases, so the speed of propagation of a disease, is needed to be known. Especially this is very important when we deal with new diseases, when we want to know how fast they are moving. And as we said, when we do a study either for prevalence or incidence, we would have a defined population, so it's the population at risk or the population under observation. And then we develop a baseline where we do a first checking of the health status. And when we do this first checking, we would find that there are some cases, and these cases are usually called prevalent cases. And if we follow through time, of course, we are interested into the new cases happening in the population, so we will follow through time the cases which did not have the disease during the baseline time. And if we follow through a year, of course, new cases will develop disease, and then those are called incidence cases. And as we said, if we calculate the prevalence for this period, we would account for both new and old cases. And if we calculate the incidence, we would account only for the incidence cases, so the new ones. The relationship between incidence and prevalence and the deaths or recovery, it's depicted in a classical analogy of a bucket, where the pebbles inside the bucket are cases. And at the baseline, let's say, we have a certain level of prevalence. And then if we follow through time, let's say, new cases will be added in. So new pebbles goes into the bucket. So it means that the prevalence will start increasing. So we would add more in terms of incidence, so an increase in incidence would be accompanied with an increase in prevalence. And if we follow through, let's say that the death or recovery rates are happening fast enough, it means that the baseline, the prevalence would decrease, because there are more cases going out or either dying or either recovering, and therefore the prevalence would drop down. But in general, as we see in figure 4 here, in general, there would be cases adding in and cases going out, and then it's, let's say, an equilibrium achieved. Of course, there is the equilibrium, a steady state, it's not achieved like a line, but normally it's fluctuating. So diseases, especially when they become endemic, they fluctuate, so they have their baseline. And if we had in mind that the prevalence is dependent on the deaths and the recoveries, of course this is linked to the duration of a disease. And because prevalence is sensitive to that, a disease in a population, the prevalence might be higher or lower depending on the average duration of the disease, meaning that how fast an animal will recover or how fast the animal will die. And usually diseases which are with short duration and are usually fatal, I mean killing very fast, like rabies, let's say, might have higher incidences than prevalences, because cases do occur, but they don't last long. So if we do want to check the prevalence in time, we would find very few cases prevailing at the time we are checking in. And while on the chronic diseases such, let's say, tuberculosis or anaplasmosis or other parasitic infections, the cases persist for a long time in the population, so they are prevalent for a longer period, and so they most likely would have an increased prevalence in such instances. But as we said, the relationship, apart from the duration, is also with the incidence rate, because, as we said, prevalence was dependent from incidence rate, that as more new cases pour in the population, the higher would be the prevalence. So the prevalence basically depends by two factors. I mean, one is the incidence, and the other is the duration of a disease. So a change in prevalence could come because of a change in the incidence rate, because either higher or lowering up, it would affect also the prevalence. A change in the average duration of a disease, so disease could be longer or shorter depending on different factors, so that would affect also the prevalence. And then a change in both incidence and duration, of course, would affect the prevalence of a disease. Normally, the relationship between incidence and prevalence is a complex one, but however, mathematically, the relationship can be derived, especially if we take as an assumption a steady state condition. So assuming that a stable population and a constant incidence rate and prevalence is there. So I mean, that's the steady state, having all measures and the population in equilibrium, that we would find a mathematical relationship into that. And the mathematical relationship is that the proportion of animals with disease above the 1 minus the proportion of animals without the disease, I mean, which is 1 minus the proportion with disease, would yield the proportion of animals without the disease. It's equal to the incidence rate multiplied by the duration of a disease, so, or let's say the average duration of a disease. But however, if the prevalence is lower than 10%, we could assume that the prevalence is almost equal with the incidence rate multiplied by the average duration of disease. If we know two of the components, we could rearrange the formula, so we would find the third component. So rearranging the formula, we would find, let's say, the incidence rate or the duration of disease. So let's see an example for the incidence rate. And we are taking as an example leptospirosis, which is a zoonotic disease, and a study showed that the prevalence of acquired leptospirosis in farmers was 34%. And in another study was indicated that leptospira persisted for 10 years, so that the average duration, let's say, of the disease was 10 years. And then the number of notified cases of human leptospirosis had remained almost the same, more or less than 10 cases per year, which means that it could be assumed that there is a stable endemic level of a disease in this human population, and of course in the cow, which is spilling over the disease to humans. And if we rearrange the formula, we would see that the incidence rate is the proportion of animals of humans in this case with the disease above the proportion without the disease, and that's all above the average duration of the disease. And we have to take into account that we are not using directly the prevalence here, but we are using the proportions because we have a higher prevalence level, so it's almost 34. So we replace the formula with the figures we had from the example, and we would find that we have 0.052 cases per year, or let's say 5.2%. So that's how we calculate the incidence rate when we had known the period, the average duration of the disease, and we had known the prevalence. The same can be applied to identify to calculate the average duration of the disease. And let's say that we have an incidence rate of rabies which was 46 new cases per 100,000 animals a year, and the prevalence in the whole population is 4 per 100,000 population. And therefore, rearranging again the formula, we would have that the average duration of disease would be equal to prevalence above incidence rate. And then replacing the values, we would find that the animals with rabies would survive, let's say, an average 0.08 years, which is approximately 1 month. So that's correct also in terms of what we know for rabies, that it's almost a month of survival, or surviving when from the time of getting exposed till the death of the animals. So that's how we calculate also the duration of the disease. But apart from that, there is also a relationship between the cumulative incidence and the incidence rate. In the cumulative incidence, time it's not used in the calculation, even though it's taken per granted, because we say the cumulative incidence, it happens for a period of time, but it's the time itself, it's not used in the calculation. On the other hand, the time, it's a component of the calculation of incidence rate in the denominator. And then incidence rate can basically be used to estimate cumulative incidence, especially taking the assumption that the incidence rate is constant and the cumulative incidence would be then equal the incidence rate multiplied by the time component we have. And especially this relationship or this formula would be true especially in populations where they are very large. And but in small population or infinite population, the approximation becomes a bit not inaccurate, especially during overtime, because the size of the population at risk decreases over time, because, and we will see, decreases over time, we will see that into an example. And let's suppose that we have a population of 1,000 cattle, which from this population, 11 cattle dies per 1,000 cattle months over a period of months. And applying the equation that we had, we would calculate that after 15 months, the cumulative incidence would be 550 deaths from this disease. But in reality, there would be only 423 deaths after 50 months of period. And because this equation basically fails to take into account the fact that the size of the population at risk declines over time. And after the first year, let's say, we would have, after the first month, we would have 11 deaths. And the population in that month would be 989, so it's 1,000 minus 11. and not 1,000. So, but the equation takes per counted that there are 1,000 animals all the time, and as a result, there is an overestimate of the cumulative incidence because the equation, it's not accounting for an exponential decay or decrease in the population of risk. So to account for that, we would use a more accurate mathematical expression by introducing a constant, which is a constant named after Leonhard Euler, which was a Swiss mathematician, and this constant is equal to 2.72 rounded. And the cumulative incidence would be 1 minus the mathematical constant at power of minus incidence rate multiplied by time. And this can be very easily calculated in Excel, and it could be that in a cell, I mean, when the function you write, you would say 1 minus exponential function per minus the incidence rate multiplied by the time. And basically that's this constant. It's used in many mathematical relationships, especially describing either a growth or a decay over time of the measures in the mathematical relationships. And if we see in this graph, I mean, there is in blue line is the calculation without the constant implemented, and then there is the improved version of the formula, which gives this slower death rate or cumulative incidence during time in 15 months. And it should be noted actually from the graph that the row or the unadapted, let's say the formula, let's say, which was incidence rate multiplied by time, would gives a reasonable estimate as long as the cumulative incidence is below 10%, which is basically 100 deaths in this case. And the equivalent, which is in this case equivalent to 100 cases because there is 1,000 population. And it would give a reasonable estimate of risk when the incidence is relatively constant, but the incidence rate, it's not usually constant in real life. So this equation is very good theoretically or under very specific conditions where the population and the disease endemic level is achieved. But in real life, that's very difficult. So there are better measures to estimate the risk, and they are basically using or estimating the risk through the survival function, which we will explain into a future lecture. So stay tuned and check that lecture if you wish to expand more on calculating risks through the survival function. Till then, be safe and see you next time.